Alright, so welcome back. This is the last portion of genetics. This is going to be the fifth time that I tried to record this, but for some technical crap, my iPad keeps crashing, so let's try this again. We are going to start learning with the following terms: diploid, haploid, locus, dominant and recessive alleles, the Punnet Square sex-linked traits, polygenic inheritance, right here, and how to spell that, Pleiotropy, I'm not really sure how I'm supposed to pronounce that, gene pool, cancer, either benign or malignant, we have tumor angiogenesis, carcinogens, oncogenes, tumor suppressor genes, and autosomes. So let's start with what a diploid cell is. A diploid cell is any cell with 23 pairs of chromosomes or 46 single chromosomes. Count them that way. A haploid cell only has half. It has 23 single chromosomes, like your sperm, like your egg. When the sperm fertilizes the egg, you finally have 23 pairs. Your locus, or your loci, for plural, is just the location for a very specific and particular gene. Your dominant and recessive alleles, your Punnet square, and your sex-linked traits. We are going to see that with, a little bit of imagery here. I want to draw a Punnet square. It's very important to know how to work with a Punnet square. On a test, you're going to get a question referring to, what percentage of boys or what percentage of girls are going to have, blue eyes or brown eyes, depending on the dominant or recessive alleles from the mom or the dad. You have to know how to work with the square first. So I'm going to draw a square here. We're going to divide it into 4 equal parts. On the top, you put 2 x's for the mother. On the column on the left, you put an x and a y for the father. You will then read and see what the mother brings to the table, what the father brings to the table. Let's just say that the mother is, let's just put 2 lowercase d's. So she carries a disease, this recessive allele, but the father does not carry this disease. He is dominant. He does not carry it, right? He overpowers that. Now we're going to take this little x and this one and multiply them. We're going to have a girl. The girl gets the dad's dominant and the mom's recessive. She is a carrier because this is another x, by the way. So this girl right here, she's a carrier because she has both dominant and recessive, but she doesn't show the phenotype because you will only show this phenotype, this disease, if there weren't any dominant alleles. So now we're going to go ahead and multiply this x with this one. We're going to have another girl, 2 x's. 2 x chromosomes indicates a girl. Now we have capital letter D, lowercase d. Same situation. Not even going to bother. Now we're going to do this y and this x. We have xy. We have a boy. If it's heterozygous, it's a boy. So we have a lowercase d and a capital D. However, we have to understand that this gene, this chromosome is gene poor. That actually means that it doesn't matter what the father has to contribute with this Y chromosome. This Y chromosome will never be strong enough to overpower this recessive allele. So it doesn't even matter that it's there. Now we're going to go ahead and take and multiply this Y with this X. Same deal. We see here that the mom gives the recessive, dad still dominating there with that gene. However, it does nothing because a Y chromosome, remember, it's gene poor. And a good way of seeing how that works is by doing this little sketch. So I'm going to go ahead and I'm going to draw an X like so and a Y like so. If you can visualize every single portion to having genetic information, right? Here's genetic information, here's more genetic information, and here's more genetic information. That gives us a total of four pieces, 4 chunks. Here's genetic information, here's genetic information, and finally here's genetic information. That gives us a total of 3. If you can picture that, if you can picture this, you'll be able to see and visualize that your Y chromosome has less genetic information than the X, which is why it does not matter. It does not matter if the father actually provides a dominant allele. If the mother has a recessive, it can't fight it at all. So that's pretty much how you can visualize that. Now let's continue with polygenic inheritance and pleiotropy. So with polygenic inheritance, we have multiple genes. So we're going to have one here, and one here. And these genes are responsible for one phenotype, may be responsible. So this gene may be responsible for brown eyes, but so can this one, and so can this one. So gene 1, 2, and 3 can all be displaying the same exact phenotype. On the other hand, if you have just one big old gene that may be responsible for multiple phenotypes, then you're looking at pleiotropy. That's that second term right here that I'm checking. Gene pool is a summation of the genotypes of a populace, cancer, benign or malignant. We're going to go over all of this in one go. So cancer, just a normal cell that tends to multiply and divide at a very, very, very fast rate compared to the other cells. So remember, what causes a cell to grow? A lot of things. Is there enough space? Are there enough nutrients? Has the DNA multiplied, right? Duplicated, sorry. Do I have enough, the growth factor? There has to be a lot of things checked off on that list for a cell to understand that it can go from G2, the prep phase, to mitosis. Cancer doesn't play by that rule. Cancer doesn't care. It just multiplies no matter what. It's benign, and I'm checking that right here. It's benign if it replicates at a relatively slow rate. Benign doesn't mean that it's good for you. It just means that it's not multiplying as quickly as a malignant cancer would. Tumor angiogenesis, and we could actually see that in this diagram of Essential Anatomy 5. It's an app. Again, I am not sponsored by this app, by this company, but I believe it's one of the most important that a student can have in order to study all 11 systems. So right now I'm just going to pop up here with an artery. And let's just say that we're going to look here at these coronary arteries. With tumor angiogenesis, I'll pick one in isolation. With tumor angiogenesis, I'm not sure if I'm pronouncing it right, the cancer tricks the body into creating and developing extra arteries, right? More arteries. And these extra cancer developed arteries steal nutrients from the surrounding, and they tend to starve the original artery. They tend to starve tissue and organs that are close to it by stealing its nutrients. That's just pretty much a very watered down version of what that is, that it might actually show up on a test. Cancer causing is carcinogens, right? You have your radiation, you have your chemicals. We can talk about oncogenes and tumor suppressor genes. Really quickly, autosomes are just the very first 22 pairs, right? So 1 through 22 pairs of chromosomes. Oncogenes and tumor suppressor genes, we are all born with a certain percentage of oncogenes and tumor suppressor genes. The oncogenes code for cancer production cells to replicate at a quick rate, where tumor suppressor genes do the contrary. If you were to be born with a higher percentage of tumor suppressor genes, you would be less likely to develop a cancer. So that's that. We went over number 1, law of complementary base pairing, right? Your A with your T, your C with your G, or if you're dealing with RNA, your A with your U, or your C with your G. You have your base triplets for your first 3, right? Your first 3 nucleotides, right? Honestly, just it's triplets, right? Just 3 nucleotides. Your codons is pretty much a nucleotide for your RNA. Your anticodon is your codon is pretty much a base triplet for your RNA, and your anticodon the transfer RNA. We talked about transcription and translation. We talked about our peptide bonds, amino acids, 12 that the body can synthesize, and 8 that you must consume. You know what a gene is. Genome, chromatin is that untangled chromosome. Genotype, phenotype, karyotype is that chart. Karyotype is literally a chart with all of your chromosomes. We learned about the chaperone protein. It's a null protein that helps the new protein bend into the alpha helix or the beta fold. In the secondary and tertiary structure, we talked about the cell cycle. Remember, your cell cycle is interphase and mitosis, further subdivides into your PMAT mnemonic of prophase, metaphase, anaphase, and telophase. You have to know what cytokinesis is. And that's pretty much that. So this is not an end-all, be-all to study genetics. This is one very small chapter of genetics in an anatomy and physiology textbook for a prerequisite course for a nursing degree. This is not genetics for a genetics major. This is extremely superficial material, very, very, very basic. It does not go in depth as much as you think it would, but it's enough to get you started for you to study on your own. You have to use your book, you have to use your notebook. My notes are not the only notes that you need for a test. Thank you.